Well, thanks everyone for coming. This is Dynamics of Human Cervical Fluid Across the Menstrual Cycle. And thank you for being here. It's really great to be able to talk to you about this. This is the disclaimer that this is not medical advice, of course, but I am sharing some fun research with you. For those of you who do not know me, my name is Nicole and I'm a menstrual health educator. I've been charting my fertility since about 2015. And I've been an artist making work about menstruation in different ways, as well as doing a lot of writing and teaching and creating other resources. So that keeps me very busy. And I just want to start off with talking about the menstrual cycle's primary purpose. Most people don't think about this when they think about the menstrual cycle. They're kind of focused on other things, maybe the bleed or perhaps reproduction. But the human menstrual cycle's primary purpose is to generate the ovarian hormones. So estrogen and progesterone perform various functions in the body relating to homeostasis and interacting with the other body systems. And they're better known for their role in preparing the reproductive system for potential pregnancy. And central to that process of fertility that's going on is the cyclical production of cervical fluid. And it's a dynamic secretion. It's made out of a bunch of different things, but you can see in the little graphic on the right that it's made up of things like glycoproteins, mucins, water is what most of it is made out of, electrolytes and enzymes. And there's quite a bit of other like cells that you'll see inside the slides as well. And if you're looking at the menstrual cycle graph above the image of the cervix here, if you follow the line of estrogen, that's the one in orange, and that's really where the cervix is getting most of its messages from as far as what to do. And it also gets a message from progesterone. That's the blue line that you see here, and it comes after ovulation. So you see both estrogen and progesterone are interacting with the cervix. And the cervical fluid undergoes significant structural and functional alterations throughout the menstrual cycle. And this has a lot to do with the crucial role that it plays in reproduction. And there's many different facets to that. It facilitates sperm transport. It provides nourishment and protection for the sperm. It selects high quality sperm. So not all of the sperm is going to meet with the egg, only a select few. And the selection is being done by the fluid. It's also a reliable diagnostic biomarker for fertility assessment. That's how we use it in the fertility awareness method. And lastly, overall, it protects the environment of the uterus from pathogens by altering the pH and the profile of microbiome that is present. And despite its pivotal role in fertility, comprehensive microscopic investigations, the structural variations and changes in cervical fluid across the different phases remain very limited. So that's the first thing that I started to understand when I got into researching. And self-reported fertility assessment of cervical fluid can be charted on a graph and analyzed with fertility awareness charting. In the case of cervical fluid, we're using a bar graph to do that. That's at the bottom of the graph. So we're actually not looking at the line graph, but instead the bar graph at the bottom. And that is how we're actually quantifying the cervical fluid across the cycle in the fertility awareness method. So in this paper, what I did was present the first public display of daily systematic analysis of cervical fluid in a human person across the menstrual cycle using the samples that I took from my body. So in the analysis is the addition of the fertility awareness chart for reference. So that is something else that I've been really curious about over my studies. And so I wanted to put it to practice. I really tried to look at the different forms of the structure of the cervical fluid and how it changed from day to day. And then I wanted to understand more about, this body of knowledge that might exist about cervical fluid and what it does and what its purpose is. So the basics of the study were that I looked at my cervical fluid every day on my microscope, and I repeated the experiment across 2 menstrual cycles. One cycle was 28 days long and one was 29 days long. I obviously didn't plan that, just how they worked out. And by analyzing cervical fluid samples collected from my own body, I tried to learn about their composition, structure, and about how much of them I was making during each day. And the purpose was sixfold, right? So I had a lot of questions. But the first thing was, let me consult about cervical fluid. It's really not that much. Characterize the features of the cervical fluid of my own and approximate the abundance of it. And then I wanted to know, what's the relationship between the types of fluid I saw and my menstrual chart. Out of all the fertility awareness charts I've made, this was a way to kind of match up what I know really well with the images that I was seeing. And then I did some comparisons and analysis of that. And I just wanted to explore what this could mean for fertility assessment, for fertile windows, potentially for the health of pregnancy. As we'll learn later, the mucus plug that you have in pregnancy, you can be at a greater risk of preterm birth or other pregnancy issues if your mucus plug is less of a barrier between you and the outside world. So the quality of that mucus plug can actually determine pregnancy outcomes. The materials and methods section, it just explained that I used this microscope that ranged from 40 times all the way up to 1600 times. So you could get pretty close up, but you could also scale back and look at the whole slide and see things from a little bit more of a bird's eye view. And they were collected using the same way that I collect cervical fluid for fertility awareness. So just using my fingers, either swiping at the vulva or swiping inside the vaginal canal if there wasn't any available at the vulva. And I just kind of went with that and checked out what I saw from there. So my eligibility here, the way that I think I made the experiment somewhat consistent, is that I have a history of regular menstrual cycles, so that I could assume that these cycles too would also be of a normal range. I didn't get pregnant through either of the cycles, and I consistently had sexual activity. Although one thing that was really interesting is that I didn't see sperm in much of my samples, even in the days after having had sex with ejaculation. So the sperm were destroyed rather quickly. I couldn't see them unless I took a sample right afterwards or something like that. And lastly, the repeatability of the sample collection. So a little bit about the history of cervical scientific study. They kind of figured out early that it was useful for agriculture. It was useful for knowing when a farm animal was going into estrus, because you could see the cervical fluid, and also for knowing when to do artificial insemination. So in the early 60s, they started to propose different types of cervical fluid and starting to categorize them in bovine samples. So that's what you're seeing here on the right. These are for samples of different cow crystallization of their cervical fluid under the microscope. But the reason why we know so much today about cervical fluid is because of the work of Eric Odeblad. Interestingly, he had a lot to do with the technology that created MRIs. He actually pioneered the introduction of nuclear magnetic resonance spectroscopy in the field of medicine. It was originally the magnetic resonance was being used in physics, and I guess the physics people didn't want the medicine people to use the device, or they were just like, we're not going to help you. But then Odeblad was like, I'm going to make my own device. And then one of the first biological samples that he started running through this device was actually cervical fluid. So during the 50s and 60s, he continued to improve the device and take more notes and study and end up publishing several papers on magnetic resonance in human tissues and mucous membranes. And these would become immensely helpful for our current understanding of human cervical fluid and the different types. And yeah, I just find that really interesting that that he was able to pioneer this. And now we use MRIs every day. Also in the 60s and 70s, you have the Billings, John and Evelyn Billings, who also were interested in this fluid, and they partnered with Eric Odeblad to ultimately come up with the Billings ovulation method of fertility awareness. So kind of with some of this scientific backing, they were able to develop their cervical mucus only method of fertility awareness. And you can see what their charts look like at the bottom here. It's just an example of what the Billings chart looks like. Here's some other existing classifications. This one was from 2014, also looking at bovine mucus. So there's basically been a couple different studies that try to create these categories for the bovine mucus. And then Eric Odeblad provided the first human cervical mucus classification system, and that's the one that I use in the study. There's also M. Cortez, who applied these fluid categories to the cow at estrus. So he took Odeblad's categorizing of the human fluid, but then applied it again to cows. And they also have a study here from N. Menarguez and Odeblad and Pastor collaborating, where they measured the range of values for each category of fluid by the cycle phase that the person was in. So we'll go through these quickly. The G mucus is what you see here on the right, these little dots. And G is for gestogenic. It's a high density of crystals appearing loose or close together in amorphous clusters with a high cellular content and tends to contain antimicrobial globulins. And this is the type that's impermeable to sperm. It creates a barrier, and that's what we see in the infertile phases. And there are two subtypes depending on if the person is in their pre-ovulatory infertility phase or their post-ovulatory infertility phase. And then a form of G mucus also makes the pregnancy mucus plug. And here's just the range of values that are used in the study, where they're categorizing a percentage of how much of that fluid type that they saw. So during the early estrogenic phase, for example, coming off of the menstruation during the basic infertile period, there's 75 to 100% G mucus. That means that, you know, basically the cervix is closed. But during the early ovulatory phase, that G mucus is 0 to 5%. So it drops from 100% to 0%, obviously, to open the cervix. And that's kind of the idea here. So we'll look at that in visual form. This is what that looks like. Basically, we're taking these values, and I just created a small graphic so you can see that the G mucus during times of high fertility is basically gone. So this is G mucus is what's associated with the infertile phases of the cycle. Now let's look at L mucus. This is more of a medium viscosity that's represented in yellow in the upper chart. That's showing you the cervix from -5 days to ovulation in the middle, 0, and then +5 days after ovulation. The red was depicting the G mucus, and the yellow is what's seen here. It's kind of like the middle and upper crypts will have the L mucus in it. And the purpose of this fluid is to attract malformed sperm or slow movers, or basically the sperm that are not genetically viable, and it's going to stop them from continuing. So it's a filter fluid, basically. It's filtering out the types of sperm that are not going to make it past the cervix. And that fluid may give a sticky sensation at the vulva when observed. So here's the L mucus. Based on the study, what they found was that it's all across the cycle, but mainly during the middle of the cycle, during the fertile periods, where you're going to see the L mucus. And here's just other examples from both human and heifer of the L mucus. Now the S mucus, S sometimes they say stands for sperm. It's a low viscosity or slippery fluid that comes from the upper crypts, which are depicted here in blue, and it helps the sperm move rapidly up the vaginal canal and into the cervix and beyond. And they can reach the S crypts of the cervix, the sperm can, in 3 to 10 minutes when it is present. So it's really like a river for the sperm. It helps move them. It also provides nourishment. It provides the channels for them to swim along, and it is a wet and lubricative sensation at the vulva that we would experience it as. And it's also can be stimulated by noradrenaline. So times of stage fright, sudden emotional disturbances, where people have a rush of fluid that's coming from the S crypts, making this S mucus very quickly. And here's that same thing shown in the graphic form. So you can see that S mucus is present really only during the times of high fertility, possible fertility and high fertility, and a little bit after. And here's just some more images of that. Okay, here's P mucus. This is the last subtype that we'll talk about, and it has several crystalline subtypes. It's fern-like, and it tends to have 60-degree angle branchings originating from a main axis. It's associated with peak day, or the last day of cervical fluid that the person experiences before their fertile window is over. And so we see it during the times of very close to ovulation, or during the peak day of the cervical fluid. And it can also be associated with rapidly decreasing estrogen levels. So here's what that looks like in graphic form. It's across the cycle, but really, again, majority towards the middle of the cycle during the fertile window is when we would see the P mucus. And P is up at the top, which makes sense because it's a fluid that helps dissolve other fluids. So the P mucus being at the top makes a lot of sense because it can dissolve the mucus plug that is below it. So P2 mucus is a well-defined main stem from which these branchings are protruding. So if you look on the right, you can see that in both the human and heifer. The P6B mucus is more like a star-like geometry or a hexagon structure. These I think are so cool to look at. They look like snowflakes or something. And again, very wet and lubricative fluid. There's PA mucus, which is yet another version of this P mucus with different sort of shapes to the crystallization. Seems to kind of go in all directions. It's really cool to look at. And then PT mucus is much more like wiry looking. It is these discontinuous crystal patterns that are dispersed and not always joined. So there you have it. That's the table taken from the data, morphological characterization of different human cervical mucus types. And we're just looking at the differences in the different phases, right? So you can see that the G mucus is clearly correlated with the time of infertility, whereas the other three mucuses show up somewhere in the fertility process, more towards the center. But the values are different for these. So I wanted to depict that a little bit as well. So you can see that in terms of actual percentages, the most is G mucus that you have, and then it, descends from there, L, S, and P. There's also the mucin polymer network, which is best observed through scanning electron microscopes. And these are the mucin polymers that bind immunological factors, antimicrobial peptides or antibodies, and bacteria. And it's responsible for the flow of the hydrogel in the cervical fluid. And these mucin polymers during the estrogenic phases have variable size pores. Basically, the pores of this mesh get wider during the fertile period and get tighter and more closed, more of like a like really fine net during the infertile phases. So a bit about Audeblad's classifications for fertility awareness. He kind of divided it by the early infertile days versus the days of possible fertility. So in the early fertile days, you'll see G minus mucus and F mucus, and these days are usually dry at the vulva for the person experiencing the menstrual cycle. The days of possible fertility there's a switch to more L and P mucus, and this is when the sensation starts for the fertile window. So we start to actually feel a moist or sticky sensation at the vulva or in the vaginal canal. And there is also some P mucus with that as well. Approaching high fertility and ovulation, Odeblad says that we have S mucus, L mucus, P mucus, and sometimes other types. There are some other types that basically they saw but weren't sure what they are. And that was the same for me in my studies here. There were many things that I don't know what I'm looking at, and they aren't classified types. So hopefully we'll learn more about that in the future. And then in the late infertile time, after the fertile window is concluded, you have the G plus mucus. So 4 days or so after the last day of cervical fluid, the person is going to have their G mucus plug set again, forming that impenetrable mucus barrier in the cervix. So I had 2 fertility awareness charts that went along with my studies here. This is what they look like. And you can see in this, let me see if I can get it to play. I actually got on YouTube and ended up putting together all of the cycle days so you can actually watch me go through the experiment the first time. So let's see, I can skip through a little bit. It's pretty fun to watch and see me sliding over some of these interesting looking forms. Now, I also did this with lactobacilli under the microscope. So I took some video of the bacteria as they were swimming around as well. The bacteria and the cervical fluid are on 2 different magnifications, so you have to like switch back and forth to see them. See if I can get it again too. Okay. And then in the study, I show my data. So what you're looking at here is the cycle day 1 of the cycle 96 and cycle 100, which were the 2 cycles that I studied. And here, same thing. So it's cycle day 15. On the left, I chose one image, and on the right, I chose another to represent some of the captures that I had. I had taken many, many captures, dozens of images and also videos. But these were some that were representative of maybe the majority of something I saw in that sample that day. Same thing. So for cycle day 21, cycle day 22, these were 2 different examples of samples that I took. And then we have the results. So the first thing that I tried to do here was to look at the existing categories and look for the angles that they were talking about and look for the shapes that they were talking about. So the L mucus, I looked for the 90-degree angle present in some of the mucus forms. And if you look at how amorphous most of the forms are, it was really interesting to see it be this perfect 90-degree angle in certain places. For the P mucus, I saw lots of the different subtypes of P mucus. So here was the P2 mucus with some of that 60-degree angles off of the main stem. Same here with the P6B mucus. I was again looking for the 60-degree angle. I was also looking for the star-like geometry radiating from a central axis point. The PA mucus, I also seem to find really strongly in this one particular sample too, that's on the bottom right. And comparing it to this heifer sample from M. Cortez, these look remarkably similar. So I thought that was really fascinating as well. And here we have the PT mucus, that wiry mucus. I did see that a few different times. So you can see that in my results on the right, comparing it again to the heifer from M. Cortez. Compare there. I also saw lots of S mucus, so I did see these lines, both discontinuous lines, like little dots in a line, and the lines, and some of the lines also had like some little organic forms like coming off the sides, which if you look at O sample on the left here, you can see a little bit of that too, where the line has these little tiny little side branches coming off of it. Pretty interesting. And then the G mucus, I also saw lots of G mucus. As you can see on the right, it's like all these little dots you see on the slide. And lastly, I did see the mucin network as well. Usually on the edges of the slides, I was able to see these mucin like net mesh pieces that look a lot different from the ferning patterns and the other types of mucus that you see. So that was it for the identification of the known types. But I also found that there were many unknown types, things that have no existence in the literature. So I'll just share with you the different categories that I came up with of what these are, what they might be categorized as. The first one is block type mucus. This mucus appeared in various forms with the fern patterns and without. And it had well-defined edges and the absence of branching, and it formed more of this blocking shape. You can kind of see it here in various forms with the arrows that I put. So yeah, I didn't know where this came from or what this might be. I also saw various salt crystals, like what literally appear like salt crystals. And because of cervical fluid's composition, it may be NaCl, KCl, CaCl2, and crystalline shapes have been observed with the salt crystals as well. So these are ones that I saw by themselves, basically, but there were also salt crystals with the ferns. So to discuss that a little bit, this crystal center fern type mucus kept coming up. This ferning mucus appeared with a salt crystal in the center, and then the fern looks like it protrudes in various directions. And oftentimes the shards of the crystal will appear to like fracture outwards in the direction of the unfolding fern, excuse me, of the unfolding fern. So if you see in the images here, there's a pretty obvious looking salt crystal there. And then their unfurling of this very organic pattern seems to go in this outward direction, particularly the one that's on the left column in the center. You can really see how it appears to be like exploding outwards, with some of the shards actually having moved along with the unfurling of the fern pattern. So yeah, this one was really cool looking. There's also ferns with orbs. So I don't know what these orbs are either. There are these round circles that I saw sometimes around the edges of a fern pattern. So you'd see these normal ferns for many days without any of these strange little, what looks like G mucus perhaps, around them. And then you see some that look almost as if they're getting like devoured, like um, by these little orbs or circular pieces. So that was really interesting to me too, which days this showed up. I also saw a lot of round ferns, or things that look like a rosette pattern. They had no discernible branches, but they unfolded outwards similarly to like how petals would unfold. And some round ferns branch outwards evenly, while some appear to have several layers. I also saw quite a bit of other glass shard looking pieces that don't resemble the salt crystals, but they're these other shard-like pieces. I also found many other ferning patterns that I just don't know what these shapes might be. Things like pinwheel shape or an X shape. Some with like two really developed ferns coming out of the X and then the other two being really undeveloped. And then I also saw a bar shape where they were just basically going off in two separate directions from the center axis. There are some that look like a half circle, some that look more like a fan. There are also K-shaped and T-shaped ones. So many different expressions of this fluid pattern. Another really interesting thing I saw was that you would see on the slide, if you went back to 40 times, so the zoomed out version of the slide, you would see multiple ferns in a clearly defined straight line across the slide. So that's not depicted in the literature either, about seeing the ferning patterns go in these straight lines like this. So I just thought, wow, this is really fascinating how all of these ferns on the slide are going in one direction. You can also see it in this one as well. It's going across in a diagonal across the slide. And here's another one going straight across horizontally. All these very cool pinwheel looking fern patterns just going across the slide right in a line. So fascinating stuff. And then I also proposed this potential for these ferns to go through a maturation and degradation, which isn't described in literature either. So when I looked at this all cumulatively, I was curious about the salt crystal before the ferning process, what happens when it explodes or expands or unfurls, kind of like popcorn does. And then the fully realized stage where the salt crystal is in the center and the ferning pattern is fully unfurled. And then the last stage I saw was there seems to be some fern contracting. So like there's almost like a dehydration of the ferning pattern. So that's what D is showing you. There's like a phantom fern outline type of thing that I was seeing. And I just thought that was really curious of, does this mean that the salt crystal is powering this fern pattern to unfurl and then eventually it degrades? So we'll have to find out more. Let's get more research on the cervix. That would be awesome. So the next thing I did was analyze those cervical fluid samples of all of the images that I took. I searched through for each cycle day. And then I try to figure out where in the cycle did those observations happen for the subtypes that are already known and unknown. So if you're just browsing on this, you can see the 4 major subtypes, G, L, S, and P, and when I saw them. So for the G mucus, very interestingly, I didn't see it during the fertile window, just like the literature says. Whereas the other types I saw more towards the center of the cycle or all across the cycle in the case of the P mucus. And then the mucin network is at the bottom. That one I saw not for the first half of the cycle, but for the second half. And the bar graph here, if it's at the 1 level, it means I saw it in one of the 2 cycles. If it's at the 2 level, it means on that same cycle day, in both cycles, I saw this mucus. So for example, in the S mucus on cycle day 11, in both experiments that I did in cycle 96 and cycle 100, I saw S mucus on cycle day 11. So you can see when it was seen on the same cycle day in both cycles. And here is the cervical fluid unknown subtypes by cycle day. So all of those different types that we were just looking at, I measured when I saw them in the cycle. So The unknown ferting patterns, for example, I saw those all across the cycle. The ferns with the orbs, I only saw during this very small window of the cycle. I think it was days 14, 17, 19, and 22, for example. So I was just trying to understand, is there a correlation here with fertility as well, or with the known subtypes for these unknown types, or do they show up more or less all across the cycle or randomly? Then I looked at cervical fluid changes and hormonal fluctuations as represented in my fertility awareness chart. So many of you know the charts observe 3 fertility signs which combine together to corroborate the fertile window. And both cycles were typical, with a fertile window consisting of a center patch of cervical fluid and increased vaginal sensation with low temperatures. And after ovulation, there was a measurable basal body temperature increase with the drying up of the cervical fluid. Round 1 had a less robust basal body temperature pattern than round 2, but both cycles were likely to have been ovulatory based on the cumulative biomarkers. And there was also a 0.2 degree difference in the cover line. So the cover line is kind of how we determine the difference between the pre and post ovulatory phases. And for the first cycle, it was 97.4. And for the second cycle, it was 97.6. And then I went and made a composite cycle just so that I could combine and you could see together. These are both individual cycle patterns, but they follow the same general menstrual pattern. So then I compared the composite cycle to my cervical fluid observations in quantified form. So for the G mucus, you can see during the fertile window, the cycle days 10 through 18 were where I didn't see any G mucus. And this happens to overlap with my fertile window in both cycles. So you can see that depicted here with the pink box. Same thing, I took the S mucus observations, which were only seen between cycle days 9 and 21, and many times seen on both cycles on the same cycle day. I took this and then compared it to the chart. Again, you can see the overlap here with the fertile window. The L mucus, this was the most difficult mucus for me to observe out of all of the types, but it correlates with the period of heightened fertility along with the beginning of the luteal phase. And then the P mucus I saw intermittently across the cycle with a trend towards more consistent presence during the fertile window. So cycle days 12 through 19 had all but 2 days with P mucus, and these days also correlate with both non-peak and peak fluid observations that I saw in my charts. So that's just represented there. And the mucin network, here's that comparison. So I saw it between days 14 and 25 in both cycles. So that's the ovulatory into the luteal phase where that was seen in my experiment. So overall, there was a clear correlation here with the sensation changes and the presence of cervical fluid and the fluid type observed under the microscope. There's also some knowledge about the cervical fluid subtypes and their impact on fertility. So I gathered up more information about those studies. Basically, they correlate with some of the same findings that we're finding about the human studies. So there's a close relationship between the mucin network and the ability for sperm to move. It helps to improve estrus detection in cows and to find the right time to artificially inseminate, to look at visual forms of cervical mucus or look at them under the microscope. People with PCOS have changes in their cervical mucus. So there was quite a bit of different studies that I pulled together of the existing literature about fertility specifically and how it connects to cervical fluid. Such as the work of Katharina Rebeck at MIT, who studied if there's any difference in mucus plug permeability between people who went into early labor. And she found that those who had preterm labor had a mucus plug with more permeability. So yeah, there's a lot there to look at. I also found a study about how the cervical fluid has really important proteins in it for defensive immune response. So those immune response proteins, some of them disappear during the fertile window, again, having to do with the letdown of the immune defense during the fertile window when the sperm would be collected at that time. And this has to do with the close relationship between the immune system and the menstrual cycle called the immunomenstrual axis. So overall, my observations underscore the complexity and variability of cervical fluid and the presence of these unique formations that we don't know what they are. They might play a role and we just don't know about them yet. So I'm trying to understand more about what these forms might mean. And understanding what these structures mean could provide more diagnostic insights for the menstrual cycle and for fertility. And some further questions that I have are, what processes drive the formation of these atypical ferning patterns? How do the variations in the salt crystal behavior correlate with the shifts in estrogen and progesterone? And could the presence or absence of certain mucus types serve as early markers for conditions affecting fertility, like cervical insufficiencies or inflammatory responses? And what role does the immunological aspect of cervical fluid play throughout the cycle? And could variations in the presence of immunoglobulins or other defensive components offer clues about the cervix's response to pathogens or response to different kinds of sperm during different cycle phases? And these were just some really cool ones. So I'm going to show you just a few fun and interesting slides of cool captures that I got over the course of the experiment. So yeah, there's just so much, there's like whole worlds inside of uh these captures. It was really, really amazing to see. And actually on my second round, on my very last day, so cycle day 29 of the second cycle that I experimented with, I saw this observation, which if you notice those really intricate, beautiful ferns, um, that we saw, they are, can I draw on this? Yeah. They were these, like this size here. And so when I came upon this thing, I was like, what on earth is this? I've never seen anything like this. So I ended up having to take several captures to capture it. It's absolutely massive, whatever it is. I don't know what it is, but it was inside of me. So that was fascinating for me. I ended up making this composite image. This is by and far the largest observation that I got throughout the entire thing. And it was on the very last cycle day. And yeah, again, here's like a normal size fern. So really wild. So that's it for the presentation. Thank you so much for coming. This is the current chart that I'm on. So I'm actually on cycle day 19 right now, and you can see that I don't have a temperature rise here yet. So I should still definitely be in the fertile window. So I'm going to switch over and we can take a look at today's sample. And if you want to stick around to go through the microscope for a minute, we'll check it out. So just give me one moment to switch us over to that. Okay, so we're back inside of the microscope. I'm gonna shut my light off. And let's see. First, I gotta get us on. Okay. Maybe 75%. All right, hopefully everyone can see that. Okay, so here we can see the basics on the slide. Looks like some cells and other things. Let me see if I can. Yeah, so these look like, these right here, your typical epithelial cells found in the vaginal canal, like what people call vaginal discharge. Just like the literal like side cells of your vaginal walls. That's what a lot of the sample will be, because I'm not taking the actual observation from my cervical crypts. Like some of the scientists, they're actually like using a little suction to like suck the type of fluid right out of the cervical crypt. But obviously I'm just doing a grabbing it from physical. So you're getting a lot of vaginal cells in here as well. So that's mainly what you're seeing here. This is like a bunch of thick vaginal cells, kind of like the, I guess like that milky, like kind of solid discharge that you would see. That's a lot of that is this. What we call in fertility awareness is vaginal cell slough, and it can be confused sometimes with cervical fluid. So we try to teach people that those two things are different. Oh, this is interesting. This is a yeast. So through the experiment, I also found that you have a lot of, there's a whole microbiome inside of the vagina as well. So you actually have a lot of different types of like commensal microbes that aren't like candida necessarily, but are types of yeasts. And let me see here. So this is like the edge of the slide somewhat. Okay, so here I'm going to switch now from sort of the cellular view of things to this, this is like the level where you see the cervical fluid in terms of magnification. So we see some like salt crystals and things here. Maybe some mucin pieces, some other stuff. And these look like potentially more salt crystals. This could be a little bit of mucin here. Yeah, that looks like the mucin network, which is always cool to see this fun mesh network. And let me see if I go up in magnification here. We can see it's even more intricate. Whoops. You can see kind of like a little bit more of its 3D nature this way. Does anyone have any questions? I'll check the chat. Typically, cervical mucus is described as egg white and stretchy. Is that the mucin network? Yes, it can be. I also believe it is potentially a mixture of S and P fluids. The literature says that the mucin network is going to open, like the size of the pores basically are going to open or close depending on the cycle phase. So it may be present, but what I found was that the exterior things that we're experiencing are probably a mixture of these, and not like one isolated from another, I think. Let's see what else is on here. Oh, going off the slide, sorry. Anything over here? Oh, what's over here? This was me every night for 2 months. What's on this side? I just would be spending like an hour on this each day. It was really quite interesting. Um, but I got used to it being like part of my nighttime routine. I was just like, okay, I gotta go do my experiments. So this looks like lots more of that. Lots of mucin present in this slide. And here, maybe these, if you can see, they're like different size little circular things. I saw these throughout too. Some of them were like fluorescent green. Some of them were different sizes. I think those might be types of immune cells, like types of white blood cells, perhaps. And when I was menstruating, I also saw red blood cells, which look exactly like what they tell you it's going to look like. So that was cool to see my own cells like that. This may be some G mucus, possibly. This looks like we've got some dots. This looks like another mucin piece. Yeah. Haven't seen any ferns on this today, though, which is pretty interesting. Some other pieces here. Let me see if I can zoom out and find some other stuff. So here we are, zoom back out again. Wow, yeah, so this slide is really dominated by like a lot of the mucin, somewhat empty in terms of the ferning. Like when I would see the ferns on the days where I would see ferns, it would be like a lot of them throughout the whole sample. Like you'd just be scrolling along and then you'd just see a bunch of them. So it's kind of interesting that I don't see much here. More big pieces. Interesting. Let's go over this way. Sorry, we got some air bubbles in here. This is more cells. Let's see if I can get, oh, there we go. So yeah, this is a pretty empty slide. It's actually a lot physically. Like I noticed I had a good amount of physical fluid there, but I don't see a lot of ferns in here. Over to the edge over here. So you can see some of these. It's kind of cool. You can compare like what the individual vaginal cell looks like compared to the cervical mucus, which is like a lot tinier. Oh, here's some. These might be in a little bit of a pattern. You can kind of see some of the S mucus looks like it maybe wanted to form here. It's kind of going across, but then it loses its shape and it's just amorphous. You can see it a little bit more here. Kind of goes in this long line. Yeah, you can see it here. So it's like, it doesn't look like the perfect lines, but you can see the general shape here is that it's following a line. And then usually what I would see is it goes all the way across, like for a long way. This one's a little bit weaker to see, but you can kind of see where the line is and where the absence of the fluid is. Go across like that. And then here's the edge of the slide. Cool. So, does anyone have any questions about the study, about the experiment, about any of the elements of the different types of cervical fluid? Happy to spend some time answering questions, or if anyone just has any general comments about this, I would love to hear from you. Thank you so much. Really appreciate you guys coming and checking it out. This was a pleasure to be able to do, and there were many things that I learned from it, and many things that I think need to still be uncovered. And I hope to continue doing more experimental research with the microscope. It's a lot of fun. So thanks for coming. Really appreciate everybody making it. And yeah, I've also found that other people are really interested in this and end up getting their own microscopes too, which is cool. There's a few other experiments I'm trying to run on this. I want to do a pre-cum experiment to figure out if, at least in the one single person that I'm having sex with, if they have pre-cum in their sperm. Excuse me, sperm in their pre-cum, because that's a question that basically when it comes down to it, people say there isn't enough research, like whenever you ask about it. So I'm just going to find out from the source whether or not it's in there. So yeah, I'm curious about that. Yes, does someone want to speak? The mic is yours.
I just wanted to say thank you so much for your time. Something that made this very enjoyable, learning about your insights, is seeing how much you enjoyed it, and making a nighttime routine out of it is necessary. So thank you so much for showing us your findings and taking so much pleasure in finding new discoveries. I can't wait till the next presentation.
Thank you so much. Yes, I am a total nerd in this way, and it only leads me to great things to be super nerdy. And this is something I've accepted about myself. So it's my pleasure to share this with you. And yeah, I hope to do more of this kind of stuff. I think it would be fun to share and explore more of the body this way. And I just think there's so much to learn from these this micro look into the world. So it's been absolutely fascinating. And if you want to see more examples of it, the YouTube has both the videos, and the videos have hundreds of examples of the different ferning patterns in full size. I really believe that you got to see this in the full screen to really to really get the look of it. But when you're scrolling over it and you're seeing these different forms, it definitely inspires. And it inspired me as an artist as well. I thought, wow, this is a medium for art. So I hope to continue making art about this as well, and kind of using these organic forms as jumping off points for some of my artwork. So thank you again, everyone, for coming. It's been my pleasure. If you want to check out the study, you can find it at learnbodyliteracy.com/studies. And the full study is there, and you can read through and understand more of the scientific experiment portion of it. And I hope to share more as well on social media about this. So thank you so much for coming, and I look forward to the next time that we get to chat.